Zoology practicals will be of 30 marks. In the zoology practicals, we are dividing the questions mainly. The first question will be identification of the given model or chart. We have to draw the diagram and label the parts. For this, it will be of 6 marks particularly. 1 mark will be for the identification, 3 marks will be for the diagram, and 2 marks will be for the labeling. The labeling of the part should be 4 to 5 labeling, that is the perfect labeling should be there. And in the charts particularly, in this first question, the chart will be displayed in front of you, unlabeled chart. For the 10 minutes actually you have to see the chart, you can draw a diagram particularly. And regarding the charts actually, 11 charts will be there in our syllabus. In the 11 charts actually, in terms of the earthworm digestive system, earthworm spermathecae, earthworm nervous system will be there. In terms of the cockroach digestive system, cockroach mouth parts, then cockroach nervous system will be there. In terms of the human being particularly, human being digestive system, human being arterial system, human being venous system, and human being male urinogenital system, and human being female urinogenital system. All these are the 11 charts particularly. In this 11 charts, the easiest one will be earthworm spermathecae. In this chart particularly, there is no labeling and all these things. Easy chart will be there. Just the diagram you have to draw, and 6th, 7th, 8th, 9th segments will be consisting of a pair of spermathecae. The parts of the spermathecae are ampulla, diverticulum. And whenever you are drawing about the earthworm digestive system, compulsory you have to remember the segments particularly 1, 2, 3 segments in the earthworm, buccal cavity, 4th segment pharynx, then 5, 6, 7 esophagus, 8th segment gizzard, 9 to 14 stomach, 14 to last intestine, 26th segment intestinal caeca. This you have to remember. And whenever you are drawing about the earthworm nervous system, the parts will be there: supraesophageal ganglia, subesophageal ganglia, nerve cord, then ganglions. All these things will be there particularly. And whenever we are coming for the cockroach digestive system, easy diagram will be there, and parts are clear actually. The esophagus and the stomach in the cockroach we are calling as a crop, and particularly mesenteric ganglia will be there, or hepatic caeca will be there, malpighian tubule will be there. All these things you have to draw particularly. In the cockroach mouth parts, upper lip, lower lip. Upper lip we are calling as a labrum, lower lip we are calling as a labium, prementum, mentum, submentum. Then all these maxillae parts, cardo, stapes, palpigere, all these parts you have to draw in the particularly cockroach, particularly mouth parts. Then cockroach nervous system also, remember the point clearly. 6th abdominal ganglia is the largest ganglia in the cockroach nervous system, right? This point you should remember particularly. And in the human also you have to draw all the diagrams, and remember the point clearly. In the charts particularly, diagram you should draw neatly. For the identification you will get 1 mark, then labeling you will get particularly 2 marks. Remember the point clearly. 4 to 5 labeling is enough. Don't write wrong labeling. Whatever you know perfectly, you have to write that labeling particularly. And the second question whatever we had in the zoology will be of the experiments. For the experiments it is 5 marks particularly. 1 mark is for the writing of the principle, and 3 marks is for the particularly procedure, and then performance, and then writing of the procedure. Then 1 mark is for the result. In the zoology actually experiments are of 5. The first one will be identification of the starch from the given sample. In the zoology experiments we have to remember the thing actually. A, B, C, D samples will be given. For all the samples you have to do the test particularly. In which sample the given, particularly in the experiment, the given sample is there, you have to test it, and you have to write the tabular form, and all these things you have to write particularly. The first one, whenever we are discussing about identification of the given starch, procedure is simple. We have to take a test tube. In a test tube we have to take the starch solution. To the starch solution we are adding the iodine. When we add the iodine we will get the blue color. That is a simple thing actually. The second one will be the demonstration of the action of salivary amylase on the starch. In this particularly, the same first experiment will be repeated. Starch sample will be taken, then iodine will be added to the starch sample, testing of the starch particularly. After that thing, salivary amylase will be added, and without disturbing it, we are keeping for it incubation. After the adding of salivary amylase, we are making the sample into 2 equal halves. Then again we are doing the iodine test. If blue color is not formed, we are going for the next step. Next step is simple in the second experiment, that is actually to the star sample, we are adding the particularly what? Benedict's reagent. Star sample, it means on which salivary amylase is acted, right? And we are adding the Benedict's reagent, we are heating it on a water bath. If the color is changed to the brick red, it indicates the principle of the second experiment, that is demonstration of the salivary amylase. Remember the point clearly. Salivary amylase will convert the starch into dextrose and maltose. That means the result you will get particularly as when we add the Bendix reagent, when you heat particularly thing on a water bath 45 degrees Celsius, you will get particularly brick red in color. Then the third experiment particularly we had about the glucose, identification of the glucose in the given sample. The same will be repeated. We are taking a glucose sample in a particular test tube, ABCD test tubes. We are adding a Benedict's reagent. Then we are heating it on a water bath. And remember the point clearly in this thing. When we are heating on a water bath, actually color change we have to see. If green color is formed, we are saying it as a 0.5% glucose. If yellow color is formed, we are saying it as a 1% glucose. If orange color is formed, we are calling it as a 1.5% glucose. If brick red color is formed, we are calling it as a 2% glucose. We are writing this result in a tabular form particularly. Then the next one will be the identification of the lipids in the given sample. Lipid sample, that means oil we are taking in a particular test tube. To that we are adding the ethanol, because ethanol is always oil is, or else lipid is always soluble in the ethanol. It is not soluble in water. When you add the ethanol, the lipid will be soluble in the ethanol. Then if you are adding a water again, the liquid droplets will be forming. Formation of the liquid droplets always indicate the presence of the lipid in the given sample. That is the fourth experiment, right? The last one will be the identification of the albumin from the given sample. This is the simple experiment. That is, in this thing, what is happening, it means actually albumin will be converted into albuminate. That means whenever we are adding the concentrated nitric acid to the albumin sample, we will get the albuminate. That means first we are taking a test tube. To the test tube, we are adding a concentrate nitric acid with the help of a dropper carefully. And remember the point, earlier 4 experiments, we have been taken a sample in a test tube, but here in the last one, demonstration of the albumin, we are taking first concentrate nitric acid with help of a dropper. Then after taking the concentrate nitric acid, then carefully we have to hold it the test tube holder. Then through the walls of the test tube, we are adding what? We are adding a sample. Whenever the albumin sample is meeting with the concentrate nitric acid, we'll get the white color precipitate ring. Formation of the white color precipitate ring always indicates what? Presence of the albumin in the given sample. And remember the point in terms of the experiments particularly, you have to write the result in the tabular form only. If the sample A it is formed, how it is formed you have to write, then hence seen you have to write particularly. Don't make mistake in the experiment particularly. 5 marks will be there for the experiment. Then the third question will be there for the spotters. The spotters is of A, B, C, D, E, F, G, H particularly. That means 7 spotters will be there. 7 into 2, 14 marks will be there for the spotters particularly. In this spotters, actually the first spotter will come from the invertebrate slides. Amoeba, Euglena, Paramecium, Hydra. Out of these 4 slides, actually you will get 1 slide. Then the next one will be invertebrate specimens. That means 2 specimens you will get particularly from the invertebrate specimens. Invertebrate specimens, it means from the phylum Sponges, you will get about the Euspongia. Then from the phylum Nidaria, you will get about the Aralia or jellyfish. You will get about the Adamsia or sea anemone. Then from the particularly what? Phylum Nematoda, you will get about the Ascaris male, Ascaris female. Ascaris male, Ascaris female is an important specimen, right? Both are the important specimens. Then from the phylum Platyhelminthes, you will get about the particularly what? Particularly liver fluke, that means Fasciola hepatica. Then about the Taenia solium, pork tapeworm, common name. Then in the phylum, from the phylum Annelida, you will get about the Nereis, sandworm or leech hirudinaria, and then earthworm, Peritima you will get. And from the phylum Annelida, particularly you will get about the Palamnios, that is scorpion, Arania, that is spider, then Palamon, that is prawn, then Scolopendra, centipedus, 100-legged, Julus, millipedus, 1,000-legged. Then insecta, in the insecta you will get about the silk moth, Bombyx mori, or Episindica, you will get about the honeybee. All these things from the phylum Arthropoda. From the phylum Mollusca, you will get about the Pilaglobosa, snail, and Unio. You will get about this both in the phylum Mollusca, and from the phylum Echinodermata, you will get about the Asterias, that is starfish. Out of all these things, you will get 2 specimens. That is invertebrate specimens, you will get 2. Slides, that is from the invertebrate slide, you will get 1. Then next about what? Next about the histological slides. That means squamous epithelium, columnar epithelium, straighted muscle, unstraighted muscle, tiers of cartilage. From all these things, you will get one slide. Then the next will be what? Next will be vertebrate slides. That means tiers of stomach, tiers of intestine, tiers of testis, tiers of ovary. Then all these things we are including in what? We are including in a vertebrate slides particularly. You will get one from the vertebrate slides. The next will be of particularly what? Vertebrate specimens. Vertebrate specimens, it means fishes will be there, 4 fishes. That is shark or scoliodon, then labio, katla, rohu, all these things particularly, sirana, all these things, 4 will be the fishes particularly. In that thing particularly here, chondrichthyes is only scoliodon. Rest of all these things are the ostrichthyes or bony fishes or freshwater fishes. Then in the snakes, you will get about the naza, Russell's viper, and then hydrophis. And in the particularly vertebrates, you will get about the rabbit, that is Arctolagus cuniculus, then about the pigeon, Columbia livia, you will get particularly from the vertebrate specimens. Lastly will be joints. 4 joints will be there: ball and socket joint, hinge joint, pivot joint, gliding joint. Ball and socket joint example will be the shoulder joint. Then hinge joint example will be the elbow joint. Then pivot joint example will be the atlas axis joint, and gliding joint example will be the this joints particularly, that is carpus metatarsal and carpus metacarpus, all these things here. That means from all these things you will get the questions from the zoology, and last will be the record. Record will be of the 5 marks. Remember the point clearly. Record is the ultimate thing. Neatness of the record should be maintained, and the external will ask the viva. And the ultimate thing is actually you have to read the record completely. And the thing clearly in the zoology is actually, in the zoology the writing part will be more. Only you are doing experiment in the zoology. Rest of the things, all the things you have to write particularly, and neat presentation should be there particularly, right? The first one will be, again I am repeating, the first question will be the chart. Out of 11 charts, you will get particularly 1 chart. Then for the chart, 6 marks will be there. Identification of the chart, 1 mark. Then diagram drawing will be of the 3 marks, and then labeling will be the 2 marks particularly. The second question will be the experiments. 5 experiments will be there. You have to write about the principle, procedure, then result. Tabular form should be compulsory in the result. 5 marks will be for the experiment. 1 plus 1 mark is for the particularly principle, and 3 marks will be for the procedure, and 1 mark will be for the result. Then specimens, again I am repeating clearly: amoeba, euglena, paramecium, hydra, 1 slide. Then invertebrate specimens from phylum, particularly sponges, to phylum Echinodermata, 2 specimens. Then particularly histological slides, 1. Invertebrate slide, vertebrate slides, 1. Vertebrate specimens, 1. That means totally 3 slides and 4 specimens will be there. Totally 7 spotters will be there. 7 into 2, 14 marks. Out of the 14 marks, half mark is for particularly identification, half mark is for the rough diagram, then 1 mark is for the identification characters. 5 to 6 points you have to write. In the zoology, compulsory you have to draw the diagram. In the botany, diagram is not needed. Right? This is about the zoology examination. And in zoology, writing part will be the more particularly, confidently, pleasantly, neatly. You have to represent the examination. 30 marks will be there in your hands. All the best. Hoping for the success in the zoology practical, intermediate practical. Right? Thank you.